All right, so what is the situation then when temperatures move below the optimum? What are the effects of decreased or lower temperatures on photosynthesis rate, and why does photosynthesis rate drop down at lower temperatures? There's a couple of reasons for this. One of the reasons is just reduced enzyme activity. This graph shows the activity of rubisco. So this is just a measure of the rate of activity of rubisco versus temperature. So like all enzymes, rubisco responds to temperature. And as temperatures decline, rubisco moves more slowly. It's less active. So what you can see is that this is actually a sort of broad survey of the rubisco enzyme across quite a few different C3 plants. And what they find is that there's sort of a broad optimum for rubisco activity between 25 and 37 degrees Celsius. And below that, it drops off relatively rapidly. So a big part of that reduction in photosynthesis rate as temperatures decline is just reduced rubisco activity, and certainly the activity of other enzymes as well, but this demonstrates that effect. And there's a fun phenomenon referred to as chilling injury or chilling effects that can be important in tropical and subtropical species, so plants that are adapted to relatively warm conditions. These effects occur sort of under cool but not freezing conditions. So maybe from 1 to 10 degrees Celsius, we see these kinds of effects. So these graphs show the effects of chilling injury or chilling effects on sugarcane. These are actually different varieties of sugarcane. And what they're showing is this is a time series during the passage of a cold front. And you can see over that sort of 1 week passage of a cold front, you can see it sort of moves from around 18 to 20 degrees C down into, 5 to 10 degrees Celsius during the depths of that cold front. But what we're seeing here is FV over FM. So FV over FM is a measure of chlorophyll fluorescence. So you remember that earlier I mentioned that chlorophyll fluorescence can be a sensitive measurement of stress on the photosynthetic machinery. And really, this is a measure of function of photosystem 2, or the light reactions. So the optimum ratio of FV over FM is around 0.8. So this would be the optimum. And what you can see is that as that cold front moves through, FVFM moves well below that 0.8 optimum. So what that's showing us is that photosystem 2 and really the thylakoid membrane function is being impaired during the presence of those cool temperatures. So what we see is that chilling injury basically is some effects on membranes that occur in plants that are adapted to warm temperatures. So we see things like loss of membrane fluidity. I'm sorry for my handwriting here. So loss of membrane fluidity, decreased enzyme activity, in those membranes at those low temperatures. So all of those would impair the light reactions of photosynthesis in these warm weather adapted plants like sugarcane. And those are sort of the effects that we see with chilling injury. So you probably wouldn't see these in temperate trees, but in things like sugarcane, you see those types of effect. And that would be another thing that causes drop off of photosynthesis in these warm adapted plants, the drop off below that temperature optimum. Another effect of temperature is frost injury. Frost injury just refers to injury to the photosynthetic machinery caused by the formation of ice within the leaves, either in the intercellular spaces in leaves, but especially inside the cell membrane of plants, and disruption or even breakage of cells caused by ice damage. So you can see the effects of frost injury here. This graph shows the effect of increasingly cold temperatures on needles of fir, both collected in the winter. So these are needles that basically have acclimated to really cold temperatures. And you can see that their measure of survival is good. Whereas needles collected during the summer and then subjected to these cold temperatures below 0 degrees Celsius, they very quickly succumb. They're basically measuring electrolyte leakage. So what they're seeing is that in these needles that haven't acclimated to cold, they haven't been frost hardened, ice crystals form inside the cells and disrupt the cells. And that causes damage to the photosynthetic machinery. Whereas leaves that have acclimated and frost hardened are not as damaged, so they don't get formation of ice inside the cells. So this is a nice micrograph showing ice formation in intercellular spaces of a leaf. So that's one of the mechanisms of frost hardening, is basically things like elevation of solutes inside cells that tends to basically act like antifreeze and protect the cells themselves here from having ice crystal formation in them, and the ice crystal formation is isolated to the intercellular spaces. So there's less damage to the photosynthetic machinery. Now this graph shows what happens. This is in Scotch pine, and it shows net photosynthesis throughout the day. This is time of day. For instance, on a day when there was no night frost the night before. And you see sort of a normal course of photosynthesis versus these days when there was variable levels of cold the night before, either a minus 6.5 degree frost event or a minus 10.5 degree frost event. What you can see is that on those days after a night frost, you get one, photosynthesis doesn't even start up until later in the day. That's basically because those ice crystals have to melt. And so there's later onset of photosynthesis during the day, but also the rate of photosynthesis is much lower due to damage due to formation of ice crystals in those Scotch pine needles. So this is an example of frost damage in Scotch pine. Now that damage can be repaired because you can see by April, for instance, that frost damage has been repaired. So those cells can repair themselves, and you can see recovery from frost injury. Another effect of cold temperatures can be photoinhibition or photooxidation. So when we have cold temperatures, that can basically slow the activity of the dark reactions. And when that happens, basically the ability of leaves to process the energy captured by chlorophyll can be reduced. And when that happens, you can have photooxidation. So basically damage to the photosynthetic membranes caused by the activation energy in chlorophyll and the inability to process that energy downstream. And you can actually get, for instance, bleaching of chlorophyll. This is sort of an extreme example here. This is a pine tree in the winter in North Florida after a cold spell. And basically that cold winter temperatures combined with bright sunlight can actually bleach chlorophyll and reduce photosynthesis rates in those plants. Here I'm measuring FVFM, chlorophyll fluorescence, and what I'm finding, I'm not showing the data, but what I'm finding is quite a decline of FVFM from that 0.8 optimum in these yellowed trees. And I find that when I drive around North Florida, especially during a cold spell, I'll sometimes see yellowing of pine trees, especially on the south side of trees that are receiving a lot of light after a cold spell. And that is a photoinhibition or photooxidation effect on net photosynthesis. So that's another effect of cold temperatures on net photosynthesis. Now, when we think about temperature responses of photosynthesis, certainly they're not static. The temperature optima of the response to photosynthesis can change. And those changes, there's sort of two kinds of changes that might occur: acclimation or adaptation. Acclimation refers to a physiological adjustment. So when we say that a plant has acclimated to temperature or acclimated to any other environmental condition, what we mean is that there are physiological changes in that individual that change its physiology in response to that change in the environment. So acclimation refers to a physiological adjustment. Adaptation, on the other hand, refers to a genetic adjustment or a genetic change in physiology. So we look at differences in species in temperature optima, like that first graph we looked at with temperate and tropical tree species and their temperature optima. That's an example of adaptation. An individual cannot adapt to a change in the environment because adaptation involves a change in genotype, a change in gene frequencies, and an individual just has one genotype. So sometimes colloquially we will say, oh, I'm not adapted to this heat in Florida, when in reality you should say, I'm not acclimated to this heat in Florida. So I just want to make clear the difference between acclimation and adaptation. One is a physiological adjustment, one is a genetic adjustment. So this graph shows a change in temperature optimum for plants grown at low temperatures and plants grown at high temperatures. So that could be either an acclimation or an adaptation response. If, for instance, this cold temperatures we're looking at plants that evolved in a cold place and comparing them to plants that evolved in a warm place, we might see that difference in optima. But if we had, for instance, the same genotypes and grew the same genotypes in cool temperatures and then the same genotypes in warm temperatures and saw this shift, that would be an acclimation response. So I just want to make sure you understand that difference. So I want to talk about a little bit about acclimation responses, and then I'll talk later about adaptation responses. This is an example of acclimation responses of temperature. So these are different seed sources of loblolly pine. So the different shapes in the graph are just different seed sources. So there's 3 different seed sources that they're looking at in this study. And then the filled symbols and solid lines here are plants that were grown in growth chambers at 25 degrees Celsius. The hollow symbols are plants that were grown at 30 degrees Celsius, and then these solid symbols with dashed dotted lines are plants that were grown at 35 degrees Celsius. And then each of those, grown in different growth environments, either 25, 30, or 35C, they then measured net photosynthesis at either 25, 30, or 35C. So we see an interesting thing happening here. Plants that were grown under 25 degrees Celsius, but then measured at 25, 30, or 35, show the highest net photosynthesis rate at their growth temperature, at 25 degrees Celsius. Plants that were grown under 30 degrees Celsius regime have the highest net photosynthesis rate at 30 degrees C. When you measure them at 25 or 35, the net photosynthesis rate is lower. And then plants that were grown at 35 degrees C experience their highest net photosynthesis rate at that temperature. So what we're seeing here are acclimation responses. Basically, plants acclimated to their growth temperature and essentially optimized net photosynthesis rate to that growth temperature. These data don't show us whether they adjusted gross photosynthesis or adjusted respiration rate, but essentially what we're seeing is that they optimize their net photosynthesis rates for that growth temperature. So that's an example of acclimation responses. And we didn't see much adaptation in this experiment. Now here's an example. This is a meta-analysis of a lot of different studies of photosynthesis in both conifers and broadleaves. And what they're showing here is a couple of different things. So on this side of the graph, panels A, C, and E, they're showing VC max measured at 25 degrees C, so measured at a constant temperature, J max measured at a constant temperature of 25 C, and respiration measured at a constant temperature. Whereas on the x-axis, we're measuring departures from a growth temperature. So were these plants grown in a warmer environment or a cooler environment? So measuring the rate of a process at some set temperature tells you whether there are physiological adjustments. And what this shows you is that across a lot of species, VC max and J max, those important parameters of photosynthesis, there was no significant change with growth environment. So basically the fundamental biochemistry of photosynthesis in terms of VC max and J max stayed about the same. Whereas respiration, as you got warmer, the base rate of respiration went down. So respiration rate actually was down regulated at higher temperatures. And what that means is that here on the right side, these are VC max, J max, and respiration at growth temperatures above and below normal. What you found was that those measures of photosynthesis went up or increased with temperature, but respiration didn't go up as much as you would expect. So they took this as evidence that there was not strong acclimation of photosynthesis to temperature, at least the photosynthetic mechanism. Now, the carbon balance, because respiration was adjusted down, carbon balance was acclimated as you went up in temperature, but the actual mechanism of photosynthesis did not acclimate, according to their meta-analysis. Now, as I dig into the literature, what I'm finding is that in more recent literature, it's really mixed. So in literature over the last 5 or 6 years, there's evidence here and there for strong acclimation of photosynthesis to temperature, but also people saying there isn't strong evidence of acclimation to temperature. So I think right now the jury is out. There don't appear to be unifying patterns in terms of whether there's strong acclimation of photosynthesis to temperature sort of universally across trees or across plants. So you can go out there and find literature in support of and not in support of sort of universal acclimation to temperature. So the jury's out. Now adaptation, certainly there is adaptation to temperature. So there's lots of data out there, but this is in red maple. Red maple is a tree species with a really broad latitudinal range. It exists down here in Florida. It exists way far north. So for instance, this shows data for net photosynthesis of a Florida genotype of red maple and a Minnesota genotype of red maple. And they were grown in a common garden. And what you can see is that at any particular leaf temperature, that Minnesota genotype shows lower rates of net photosynthesis compared to the Florida genotype. And certainly at higher temperatures, you get higher photosynthesis rates in that Florida genotype. So this shows us that there's definitely adaptation to different temperature regimes in red maple. And you would see similar things happening in other species of trees, especially species with really large latitudinal gradients. So definitely adaptation to temperature exists. Finally, there's some experiments out there that are looking at not just sort of steady state changes in temperature, but also looking at things like heat waves. Now this is a really complicated graph and I'm not going to go into detail on it, but basically this graph summarizes some experiments in loblolly pine and red oak where they looked at heat waves. So there's a lot going on here, but basically circles and squares show both ambient and elevated CO2. Green symbols are ambient temperature, and then ambient plus 3 degrees C is yellow, and then orange and red show plants subjected to either 6 degrees C or 12 degrees C heat waves. So basically really elevated temperature for several days. It's a big mess here and there's lots of variation, and I encourage you to go look at the papers. But basically what they're showing is that the effect of these elevated temperatures is somewhat mitigated by elevated CO2. So this dotted line versus the solid line shows elevated CO2 at these different temperature regimes versus ambient CO2 at these different temperature regimes in loblolly pine and red oak. So what you can see is that while there are generally negative effects on net photosynthesis of these heat wave or elevated temperatures, that to some extent those negative effects of temperatures are mitigated to some extent by elevated CO2. So the take-home message to me is that when we look at the effects of temperature expected with global climate change, those will likely interact with elevated CO2. And we need to look at the interactive effects of both temperature and elevated CO2 on photosynthesis and carbon balance. So just looking at changes in one factor is not sufficient. We need to look at interactive effects of, for instance, elevated CO2 with temperature in all of these global climate change studies.